Hello. This video is about the biogeochemical cycle of arsenic with a focus on the methylating process. The arsenic cycle is important because of the inherent toxicity of arsenic compounds, which makes tracking them throughout the global environment useful. Among interests are the concerns on their influence on microbial diversity, as arsenic has been employed as antibiotics by bacteria to dominate over weaker bacteria, especially the trivalent methylated arsenic species, such as the monomethylated arsenic, or MAS, and the dimethylated arsenic, or DMAS, both of which are precursors to several other arsenic-based substances. The global arsenic cycle also accounts for arsenic consumption by humans, which can be dangerous if the concentration reaches high enough levels. To discuss methylation of arsenic, some common abbreviations need to be addressed. The most important is SAM, which stands for S-adenosyl methionine. SAM is the molecule which is believed to be responsible for the methylation of arsenic in both humans and bacteria. The molecule consists of an adenosyl part and an amino acid similar to cysteine, called homocysteine, connected at the sulfur with an additional methyl group bound to the sulfur as well. Once SAM has methylated an arsenic compound, its demethylated form is called SAH, or S-adenosyl homocysteine. Arsenic methylation is encoded in humans in the gene AS3MT, which encodes the enzyme arsenite methyltransferase. A similar gene is encoded in microbials in a gene called ArsM. Both enzymes encoded by the genes are believed to use the SAM molecule for the methylation itself. Another important abbreviation is TRX, which stands for thioredoxin. These are proteins present in all organisms. They are responsible for stabilizing systems through redox reactions, and are important in the methylation of arsenic. It's drawn here as a circle with two HS sticking out. Moving on to the methylation itself, arsenic generally enters cells as arsenous acid, upon where the hydroxide substituents are rapidly replaced with a glutathione, represented as SR, while still remaining a trivalent arsenic compound. This substance then binds the arsenic to the 3 cysteine sulfurs of the ArsM enzyme. The methylation step is a simple SN2 reaction between the lone pair of the arsenic and the SAM, like so. Hydroxide reduces the arsenic enzyme molecule, and arsenic is left with a methyl group and a lone pair again, while 2 cysteines are bonded together in a mechanism like this. TRX is responsible for protonating the cysteine sulfurs, removing the bonds between the sulfur atoms. At this point, the arsenic can either be cleaved from the enzyme through TRX and water in a multi-step reaction, leaving trivalent monomethylated arsenic, which we will call MAS. Or it can get methylated again through SAM in a similar SN2 reaction as the first methylation, making dimethylated arsenic before being cleaved from the enzyme by TRX and water, leaving a trivalent dimethylated arsenic substance, which we will call DMAS. From DMAS, there are pathways to several different arsenic compounds of interest. The arsenic can, for example, be methylated again by SAM, making trimethylarsine. Oxidation in air is also a common pathway for the methylated arsenic substances, oxidizing the arsenic to a pentavalent, either mono or dimethylated arsenate. DMAS has been shown to be a precursor for arsenosugars. These are also suspected of being made using SAM, but instead of an SN2 reaction, we expect a radical reaction, using an enzyme called ArsS instead of ArsM. The bond broken in the SAM molecule is between the sulfur and the carbon in the adenosine part, and is done by a protein called ferredoxin. The following reaction between DMAS and the now radical adenosinoid results in an intermediate with a radical being located on the arsenic atom. The intermediate reduces the ferredoxin, regenerating it, and generating a double bond to the oxygen. This molecule is called 5'-deoxy 5'-dimethylarsinoyl adenosine, or just DDMAA for short. DDMAA can further be hydrolyzed to get rid of the adenine group and ending up with an arsenosugar. Arsenosugars are way less toxic than the trivalent methylated arsenic species, and are precursors for both arsenobetaine and arsenolipids. However, in humans, arsenosugars are broken down to DMAS, which is more toxic and thus unfavorable. Arsenobetaine is found in every microbial environment, and even in humans. Their biological function is not yet apparent, other than being less toxic than other arsenic substances. There are a few pathways suggested for arsenobetaine formation, either through degradation of arsenosugars or through oxidation of DMAS. Finally, Arsenolipids are another group of methylated arsenic compounds, which have been detected in fish. These include hydrocarbons, fatty acids, phospholipids, fatty alcohols, and so on. Pathways for formation are not clear, but we do know they also get broken down to DMAS in humans.